Hello students, how are you all? I hope you all are fine. Today we'll see Standard 7th Subject Science. In that, chapter number 5, Acids, Base and Salt. In the previous video, we have seen two parts, part 1 and part 2. Today we'll see part 3. In the previous video, part 2, we have seen up to main 6. Today we'll start from main 7. See, write 2 points of differences. Distinguish between 2 marks each. Difference between acids and base. See the number 1. They are short to taste. Acids are sour to taste, and base they are bitter to taste and soapy touch. See, next point is they turn blue litmus paper red, and in bases they turn red litmus paper blue. See, remember it, acids turn blue litmus paper red and bases turn red litmus paper blue. Now see third point, they remain colorless with phenolphthalein solution. They remain colorless with phenolphthalein solution, and here they give pink color with phenolphthalein solution. Bases give pink color with phenolphthalein solution, and in acids remain colorless with phenolphthalein solution. Now see the number 2, difference between acids and a salt. Point 1, they are sour to taste. Acids are sour to taste, and salts, they are salty or astringent to taste. See, next point, they turns blue litmus paper red, and here mostly they do not affect the litmus paper. Salts do not mostly they do not affect the litmus paper. Now see the difference between bases and salt. They are soapy to touch, and here salts, they are not soapy to touch. Number 2, they turn red litmus paper blue. Bases turns red litmus paper blue, and here mostly they do not affect the litmus paper. Salts mostly they do not affect the litmus paper. Now see the next page. Main 8 classify. It carries 4 marks. Classify the following substance into acidic, basic and neutral substance. First of all, we will see the given substance. Now see, orange juice, curd, soap, common salt, lemon juice, baking soda, nitre, vinegar, sugar, milk of magnesia, glucose, washing soda, turmeric solution, distilled water, flake lime and ammonia. Acidic substances are orange juice, curd, lemon juice, vinegar, turmeric solution, and basic solutions are soap, baking soda, milk of magnesia, washing soda, flake lime and ammonia. And neutral substance, common salt, nitre, sugar, glucose, distilled water. Now see main 9. Answer the following questions in a detail, 4 marks each. Describe the following experiment with the diagram. Now first of all we will see the aim. To study the neutralization reaction between acid and a base. Here apparatus and a materials. Test tube stand, dropper. Now substance, dilute hydrochloric acid, dilute sodium hydroxide, solution and phenolphthalein. See the figure. In the figure, this is the test tube stand. Here it's the test tubes are there, and in the one test tube dilute HCl, dilute hydrochloric acid is there, and in the another dilute NaOH solution is there, and in the another test tube phenolphthalein solution is there. Here in the one test tube, it's a holder is there, test tube holder, okay, dilute HCl. Test tube is there. In that dilute NaOH solution you have to add it, and with the help of dropper, drop wise you have to add phenolphthalein solution. Now see the procedure. Take a test tube, fill 1/4 of the test tube with a dilute hydrochloric acid using the dropper. Now add 2-3 drops of phenolphthalein. I have tell you, no, you have to add phenolphthalein with the help of dropper. So now add 2-3 drops of phenolphthalein into the test tube. Shake the test tube gently, no change colors in the solution. Now add dilute sodium hydroxide solution drop by drop. See, drop by drop to the dilute hydrochloric acid in the test tube using the dropper and go on stirring the test tube. Stop adding sodium hydroxide solution when the solution in the test tube turns just like pink color. Insert blue and litmus papers in it. See the next page and observe. Now see the observation. There is no effect on the litmus papers by the solution. So obtained in the test tube. Conclusion is that by chemical reaction between acid and base, neutral substance having properties other than that of acids and base are produced. This reaction is the neutralization reaction. There is no effect on the litmus papers. So this solution is the neutralization reaction. This reaction is known as the neutralization reaction. Now activities. To verify whether the given various solutions are acidic, basic or neutral with the help of red litmus paper and blue litmus paper. Now see the litmus test of the given substance. Litmus solution, test solution, effect on red litmus paper and effect on blue litmus paper and inference. Number 1, lemon juice, no effect on litmus paper, and here blue litmus paper turns red and it's a acidic. No effect on red litmus paper. Okay. Number 2, tap water, no effect on red litmus paper, no effect on blue litmus paper and it's a neutral. Number 3, detergent solution, red litmus paper turns blue and no effect on blue litmus paper, so it's a basic. Now see aerated drink, no effect on red litmus paper and blue litmus paper turns red, so it's a acidic. Soap solution, red litmus paper turns blue and no effect on blue litmus paper, so it's a basic. Now shampoo, no effect on red litmus paper and blue litmus paper turns red, so it's a acidic. Now common salt solution, no effect on red litmus paper, no effect on blue litmus paper, so it's a neutral. Now sugar solution, again no effect on red litmus paper, no effect on blue litmus paper, so it's a neutral. Vinegar, no effect on red litmus paper and blue litmus paper turns red, so it's a acidic. Baking soda solution, red litmus paper turns blue and no effect on blue litmus paper, so it's a basic. Milk of magnesia, red litmus paper turns blue and no effect on blue litmus paper and here it's a basic. Washing soda solution, red litmus paper turns blue, no effect on blue litmus paper, so it's a basic. And last one, lime water, red litmus paper turns blue and no effect on blue litmus paper, so it's a basic. Now see the next page. To prepare turmeric paper, apparatus and materials, turmeric, water, blotting paper. See, blotting paper is like this square papers, okay, and filter paper means it's a filter paper and dropper. Substance, given solution, turmeric paste and here it's a turmeric paper. Procedure, take a tablespoon of turmeric powder, add a little water and make a paste. Now apply turmeric paste on blotting paper and allow it to dry. Now next step is cut thin strips of yellow paper due to turmeric paste obtained. Now put a drop of soap solution on the strip of turmeric paper. Similarly, taste the remaining solution from the given solution using turmeric paper and note your observations. Now see the number 3. To prepare the indicator of China rose, Goodhull flower. Here apparatus and materials, China rose, flower, beaker, hot water and dropper. See here it's a beaker, hot water in it and petals of China rose are there. Procedure is collect some China rose means Goodhull petals and keep them in a beaker. Add some warm water to it, keep the mixture for some time till water becomes colored. Use the colored water as a China rose indicator. Add 5 drops of this indicator to shampoo dilute solution. Note what is the effect of indicator on the solution. Similarly, test the remaining solutions from the given solution using China rose indicator. Now see the fourth one. To study the effect of acid and base on litmus paper, turmeric paper and China rose solution. Apparatus and materials. Blue and red litmus paper, turmeric paper, China rose solution and dropper. Substance given dilute acid and base. Now procedure. Put 2 drops of dilute hydrochloric acid on each blue litmus paper, red litmus paper and turmeric paper and add the same in a China rose solution with the help of a dropper. Check the effect on them. Similarly, taste the remaining dilute acids and base. Now see the effect of acid and base on various indicators. See the table, number, name of the acid and base. Now effect on blue or red litmus paper, effect on turmeric paper and effect on China rose solution. Dilute hydrochloric acid. Blue litmus paper turns red and no effect on turmeric paper, and here China rose solution turns into magenta. Dilute sulfuric acid. Blue litmus paper turns red, no effect on turmeric paper and China rose solution turns into magenta. Dilute sodium hydroxide. Red litmus paper turns blue and here turmeric paper turns red and China rose solution turns into magenta color. Ammonium hydroxide. Red litmus paper turns blue, turmeric paper turns red and China rose solution turns into green color. Calcium hydroxide means lime water. Red litmus paper turns blue, turmeric paper turns red and China rose solution turns into green color. Now see the conclusion. Conclusion is that acid turns blue litmus paper red and basis turns red litmus paper blue. See, whatever solution is acidic in nature, that converts blue litmus paper turn red, and whatever in basic solution, that converts red litmus paper blue. Acids do not affect the turmeric paper, but basis turns turmeric paper red. See, when the solution is acidic, then there is no effect on turmeric paper. But if the solution is basic, then this turmeric paper turns red. Now see, acids turns China rose solution magenta and basis turns China rose solution green. Acids turn China rose solution magenta and basis turns China rose solution in the green. Here your chapter is completed and we will see the next chapter in the next video. Stay safe, stay healthy. Okay, bye.